All right, we're going to finish up our DNA unit talking about mutations. So, mutations, we can say that they're unintended changes. So they're unintended changes in DNA, right? So mutations change genetic information of the cell. That's what it says over here. Right. There are different types of mutations. They're going to be point mutations and chromosomal mutations. But the important things that mutations do is they drive what we call macroevolution. Right, so we're going to talk about that much later in different chapters. Macroevolution is evolution at the species level, right? So microevolution, so we'll talk about evolution coming up, but evolution is simply just the change in gene frequencies over time, right? So the way we do random fertilization and random assortment of chromosomes, that we'll talk about when we talk about meiosis, leads to microevolution, right? But macroevolution are these distinct changes in DNA. So, for the most part, mutations are not healthy for an organism. Most of them do not lead to viable offspring or offspring that are going to live. So most mutations are what they call silent. So remember, you have all of these NTRs within your DNA. So remember, we said that up to 95% of your DNA are non-translated regions, or these regions that don't code for genes. And one of the functions of all of that DNA is it's able to absorb lots of mutations without affecting the species. Right? So let's start talking about them. So we have large scale mutations. So deletions are simply, and all of these generally occur during meiosis, which we kind of flipped around and we'll talk about in a little bit. But you see that our gene sequence should be A, B, C, D, E, F, so that's our normal gene sequence. In a deletion, I just lose a gene. Now, generally speaking, that's pretty bad for an organism. Good thing for you and me, we're a diploid species, right? So if you think about it right over here, we're a diploid species, we have two of each chromosome. So if I'm missing a B in one chromosome, I still have it in the other. So I'll have half of my gene product, but for most circumstances, half of that product is good enough to get by. In some instances it's not, but in many instances it is. So deletions, inversions. So an inversion is it's flipped. Now the problem with being flipped, so if you look at the inversion here, right, we've got a loop and then it's flipped. Instead of being A, B, C, D, E, F, it's A, E, B, C, D, F. So we've got a section that was flipped, right? It's flipped right here, right? Or actually, it's flipped right here, right? So it got flipped over. So the problem now being is if I had this and I flipped this region in here, right? Now I would have right? And if you look at it, these are not the same, right? So all of my gene products would be wrong, right? So that wouldn't be good. We have translocations, right? So translocations are probably the most common survivable chromosomal mutation for our species. And really what happens with the translocations during a process called crossing over, which we get to shuffle genes on chromosomes, shuffle maternal and paternal genes, usually translocate or crossing over is what they call reciprocal, or they get the same. But if we've got uneven crossing over here, you end up with a longer and a shorter chromosome. Now, if you're an organism and you have this happen, you still have all your genes, right? The problem is now that you're going to make gametes that have more or less gene products. So they think that although a translocation in my body might be okay, when I make gametes, right? Gametes are like sperm and egg. Then we get some interesting gene combinations which may not be viable with life. They think that might lead to some infertility. A polyploidy is an entire set of chromosomes, right? So haploids or gametes, right? So our gametes are haploid, right? Our sperm and egg are haploid. And we join two haploids and we get a diploid. So this is normal for our species. And for most animals. If you get an extra set, you'd be triploid. If you get an extra two sets, you'd be tetraploid. So the important thing is plants are the only type of species that really survives these increasing numbers of chromosomes. For instance, a strawberry is an octoploid species. It has 8 sets of each chromosome. Somy, right, means body. So trisomy has 3 of 1 chromosome. So trisomy 21, which we call Down syndrome, has 3 copies of chromosome 21. And we'll talk about that a little later. So a difference between ploidies and somies. Somies are 1 extra chromosome. A ploidy is an extra set of chromosomes. All right, point mutations. So point mutations are probably the most common survivable type of mutation. So a point mutation is a single nucleotide. Right? Not a gene or a chromosome segment, but a single nucleotide. So I'll say that each carries with it the ability to alter the phenotype of the individual, right? And from the cells that create gametes, all the offspring of the individual. So most cancers are caused by point mutation, right? So a lot of our survivable diseases, right, our genetic diseases are caused by point mutations. So when the phenotype is the outward appearance, right? The genotype is the gene sequence. The phenotype is the outward appearance. So we have a couple, right? We have base pair substitutions. So there's 3 different types here. A silent, right, gives you the same amino acid. A missense gives you a new amino acid. And a nonsense, right, gives you a stop, right? So those are our 3 types, silent and same, right? So if you look at here, I'll give you an example here. So over here, right, ATG is my DNA sequence. My RNA sequence would be UAC, and I'd get tyrosine, right? If I change this G here to an A, right, I still get tyrosine, right? So that's what's called a silent mutation, because the mutation really doesn't affect the phenotype of the organism. The genotype changed because the nucleotide sequence changed, but the phenotype doesn't change, right? This one here, if I change the G to a C, I get UAG, and that is a stop, right? Since it's a stop, that would be a nonsense mutation. The problem with a stop is it stops. So you get an incomplete protein. And that protein, if it's incomplete, won't fold correctly, and it won't work, right? And the third one, the original A got changed to a G. Because of that, I got histidine, right? And I'll get a faulty program because the protein won't fold correctly, right? So that is a missense mutation, right? Changes to a new amino acid. So make sure you know the 3 different types. Insertions and deletions are the worst of the point mutations, right? And the reason why they're the worst, if I get an addition or a deletion of a a single nucleotide, it changes everything downstream. So if you see our original sequence here, TAT, TGG, CTA. So if I get and if I insert a C right here, right, then it pushes all of my codons off by one. So now it changes every amino acid downstream, right? So I get a completely different new program that won't work, or protein, not program, but a protein that won't work, right? So insertions and deletions, exception. Except if they occur in introns. Because remember, introns are non-coded regions that are going to get removed from the pre-mRNA when you make mRNA. So if it happens in an NTR, if any of these point mutations happen in NTR, it doesn't matter. And that's the beauty of NTRs, right? So that's the beauty of introns. Now, if you're bacteria, you don't have introns, and any of these kills the bacteria, but they don't care, right? Because bacteria replicates every 20 minutes. You can kill all the bacteria except for one, and within an hour, there's just a ton of them, right? Within 24 hours, there's millions of them. So that's why bacteria survive mutations so quickly and they mutate so rapidly. All right, so that was just quickly through mutations. We're going to go through them again. We're talking about mutagen causes mutation. So this is an agent that causes mutation. Right. So you can have physical or chemical. Physical, an example would be radiation, right? So UV, X-ray, and gamma. Gamma being the most harmful. And ultraviolet, right above the visible spectrum, that's what you get. It gives you sunburn, and then if you get too much of it over time, it can cause skin cancer, right? And remember, cancer is caused by these point mutations, right, in your DNA. And then chemical. So a chemical mutagen is called a carcinogen, right? So they say cigarettes have many carcinogens, and that's why cigarettes cause cancer. So they're chemical. If the agent causes birth defects, it's called a teratogen. Right? And if you look at this frog here, frogs are called sentinel species because they mutate so rapidly. Look at all the legs that frog has, right? So it's come into contact with either carcinogens or teratogens or both, right? And that's why it's like that, probably more of a teratogen. All right, so we'll talk about the Lac Operon theory. So, let me get a drink here. You have lots of resources, right? So we can make proteins all day long. We can rest and repair. We can store energy. But if you're a single-celled organism like bacteria, right, you wouldn't want to transcribe. You wouldn't want to use your limited amount of energy for making gene products and proteins. So an operon, right? This is a way. So this is the way bacteria can turn on and off a series of genes, right? So usually it works back with what they call feedback inhibition. So the product of a gene sequence then turns off a series of genes. So what it says is we've made what we need to make, so then we'll go ahead and turn it off, right? And then more substrates will turn it back on. All right, so let's look at an operon. So there's a couple different important parts here, right? So remember that transcription starts at the promoter. And if you're a eukaryote, you have that ta-ta box within the promoter that your transcription factor is attached to. But bacteria doesn't have that ta-ta box. But If we have an operon, right, within the promoter is the operator, right? So the operator is the switch. So operator is where the repressor attaches, all right? So the repressor, this is in red over here, so the repressor here, right, blocks the attachment of RNA polymerase. So what the job of the repressor is to make it so we can't try to transcribe those genes, right, unless we have something those genes would work on, right? So usually these operons block enzymes that code for proteins that digest food, right? So in this instance, lactose sugar, right, is going to be the inducer, right? And the inducer over here inactivates the repressor. So if there's lactose, right, lactose will bind to the repressor and it'll change its shape. It'll make it fall off of the operator, and then RNA polymerase can attach and make a gene product. Now that gene product will be an enzyme that digests lactose, so it will come digest the lactose off the repressor, and the repressor will reattach to the operator, right? So as long as there's lactose available, it'll keep disattaching the repressor, and then the RNA polymerase can attach at the operator within the promoter, and it can make those genes. As soon as there's no more lactose, it'll stop making enzyme, right? Because that repressor will attach, it'll wait for some more lactose to be found. Right, so three things. Know what the promoter is, right? Know that the operator is within the promoter, and the operator is where the repressor attaches. The repressor blocks RNA polymerase, and without an RNA polymerase, I can't transcribe those genes, right? And the inducer inactivates the repressor by changing its conformation. So eukaryotic regulation, we don't have operons. We do it by packaging. So this is through packaging. Around proteins called histones. So we package DNA around proteins called histones. If the DNA is wound around a histone, then RNA polymerase can't get to it. Right? If it's loose through the middle, it can get to it. So I'm not going to test you heavily on this. We'll probably get to this a little more later. So regulation of chromatin. So all these little blue kind of ball-looking things are called histones. So these little things right here are called histones. Right? And DNA is attracted to it, it winds around histones. Right? If I get a group of histones like this, so this is called a nucleosome, right? And if I get all of this together, this is called chromatin. Right? So the non-condensed form of DNA is called chromatin. So regulation, so histone modification. So acetylation, right, makes chromatin less dense, and that promotes transcription. So acetylation would cause that. So it exposes that area of DNA, and because it's exposed, then RNA polymerase can get to it, it can open it up, and it can read it. DNA methylation inactivates DNA, right? And it causes it to wind a little more tightly, right? And this right there would be an example of that. Since it's wound tightly, then RNA polymerase can't get in and transcribe it. And the reason why that's important is every cell in your body has the same DNA, But different types of cells in your body have to do different types of functions. They need different types of proteins. So since all the cells have all the DNA to make all the proteins in your body, it's got to be packaged differently so that you only expose the type of DNA that make the type of proteins that you want for that type of cell. Right? Good thing for you, there's a thing called genomic imprinting, which means if I have a cell that's already specialized, when it reproduces, it'll come up with the same packaging properties. Okay, so that's it today. It's just a brief introduction, and that's about it. Talk to you soon.